No, it was just a bunch of guys. Yeah.
All right. Good afternoon. Welcome to the second seminar for the Storer Lectureship. And today's our speaker is Dr. Charles Rice, and I'm honored to actually introduce Charlie Rice. Charlie actually graduated from UC Davis with a bachelor's in zoology. And when I first heard about him, somebody told me that he has 5 majors when he was in UC Davis, which obviously he said is not true. Yep. So he graduated here in 1974. He then moved on to Caltech to do a PhD in biochemistry. And his focus was virology. He mostly worked on Symbis virus and yellow fever. After he graduated in 1981, then he stayed in Caltech for a few more years until 1986, where he actually got his first faculty position in Washington University in St. Louis. Let's see. And then in 2000, Dr. Rice then moved to Rockefeller to be the executive and scientific director of the Center for Hepatitis C Virus. And he's also the Maurice and Corian Greenberg Professor at Rockefeller University. So obviously, Dr. Rice is it's one of the most prominent virologists in the world, I would say. And he is a former president of the American Society of Virology. He is a member of the National Academy of Sciences, and he received a very prestigious prize just last year, 2013, the Doornbende Prize from the Belgium Royal Academy for Medicine in pathophysiology for his work in HCV. It's safe to say, I think pretty much, that Dr. Rice's work has directly or paved the way to the development of the effective antiviral drugs that we have for HCV today. Besides his research, I think a lot of his contribution is also in mentoring. So over the years, he has actually trained many, many students and postdocs, many of whom I know, who went on to become very successful scientists and heads of their own laboratories in both academic institutions and also biotech companies. And just, I think, for this visit, I guess, for all the junior faculty, the postdocs, and the students who have actually met with Dr. Rice, we really appreciate your time. We really want to thank you for your advice and wisdom. And so with that, I'm going to let Charlie take the floor. He's going to tell us a little bit about HCV, which he says there's still a lot to be done, even though we have some good drugs now. And, okay, all right.
All right, well. I want to start by, thanking everybody for, coming out again, and particularly Joanna. I think she has been just an incredible sort of host for this visit, and I really appreciate not only the invitation, but the fact that she's basically taken the last couple of days off to make sure that I don't get lost, because this campus doesn't bear too much sort of resemblance to the one that I left 40 years ago after being an undergraduate here. So, I don't know how many of you were at my talk yesterday, but I sort of, in that talk, talked about sort of the sort of history of, hepatitis C and the discovery and some of the progress that's been made over the last 25 years to the point where we're looking at treatment regimens that, if sort of used sort of properly and if people can afford them, have 95% cure rates for hepatitis C. And I guess this isn't a sort of a common occurrence in sort of biomedical science, where you're in a field sort of working on a human disease where your field is in danger of being driven to extinction by sort of the success here. Uh-oh, what do we got?
Sorry about that.
That's okay. What are we?
I just need to turn the lights on just a little bit.
More lights? Oh, I don't know. People are already snoozing, sort of peacefully here. So, I guess the question sort of arises, why should we continue to study this virus? And, I think that, as with many and essentially every area that people are interested in, and HCV is no exception, there are still a lot of questions that we haven't answered, both in terms of the molecular virology, the host interactions, and sort of the mechanisms that the virus uses to persist, and the pathogenesis of liver disease. We've made a lot of progress on coming up with new therapies, but I think in terms of understanding the natural history and the progression of liver disease and HCV, still a long ways to go. This is something that's near and dear to my heart. The fact that we've spent the last 25 years trying to establish good experimental systems and animal models to study this virus. It would be kind of a shame not to use them to continue. And that includes, I think, much better and more relevant cell culture models, some of which I talked about yesterday, hepatocyte models. And because there's been so much interest in the biotech industry in terms of developing new drugs to treat hepatitis C, we have one of the best collections of chemical tools targeting almost every sort of viral function to use to try and understand how these fit together in the virus life cycle. And I think beyond that, it's very important to maintain basic research diversity. And we can't really get too focused or too translational, because otherwise we're not really going to have the pipeline of research that leads to our ability to deal with unexpected challenges when they arise. And as I say, for hepatitis C, you never know what's coming next. As I mentioned yesterday, we really didn't have any idea sort of what the sort of global distribution was of hepatitis C-like viruses, and now they're being found in horses, and dogs, and rats, and mice, and in bats. So there's a lot of stuff lurking out there, and I think our ability to sort of predict, sort of which of those might become either a veterinary or a human disease problem is really very low. So I think the playing field has changed. I mean, one always has to try and address important fundamental questions. I think the best research comes from curiosity sort of driven research, and that isn't necessarily something that can be mandated by NIH. It really has to sort of come from the individual. We got to maintain high standards. I think that's always good, and I think it's always important to apply the knowledge that we've gained from sort of one investigation to others. And I think a lot of the progress that was made in the HCV field by people that decided to enter that field had to do with sort of their prior experience working in various model systems that might not have been viewed as being all that important in terms of biomedical need. And I guess the thing that we sort of face in HCV research is, how's it going to be perceived by our peers? Are people on study section going to look at an HCV grant and say, oh, game over, you shouldn't be studying this virus anymore? But I think if you look at the advances that have been made studying, other viruses, if we hadn't been studying mouse hepatitis virus, for example, we wouldn't have been in a very good position to sort of make rapid progress on SARS and MERS. There are lots of examples like this, so we'll see what happens. So I wanted to sort of take the opportunity to tell you sort of 3 stories that are ongoing in the lab. So the first of them is a fairly sort of HCV-centric story, and this is sort of a dirty little secret of HCV research, and the fact that unlike HIV, where you can take a primary isolate and put it in a susceptible culture and grow the virus and maybe look and see if it's resistant to antiviral drugs, we can't do that for hepatitis C. So I'll tell you a little story that sort of bears on that. And then I'm going to tell you a cautionary tale that has to do with a search that we did to look for host factors that were important in HCV replication. I thought this would be perhaps a sort of interesting sort of lesson for the graduate students and postdocs. And last, I'll tell you a little bit about our work on interferon, which we got interested in understanding a little bit more about because this was the mainstay of HCV treatment for more than 20 years. And I guess at one point in the development of new strategies to treat HCV, we thought that it might be important to figure out how something that actually worked. And so that got us into that area. So this first story, I'm going to just go to backtrack a little bit. Some of this is going to be redundant from yesterday. As I mentioned, the virus was discovered by Mike Houghton in 1989 using an antibody-based screen to identify a E. coli expression clone that expressed part of the genome. And this basically identified the virus, and this was really a great boon for the development of diagnostics and cleaning up the blood supply. And this virus plopped into the family of flaviviruses, a group of viruses, of positive strand RNA viruses that we had been studying. So it wasn't until 8 years later that we actually managed to make the first infectious molecular clone for hepatitis C, and that's basically done by making a cDNA copy of this RNA virus that can be propagated in E. coli, and then using a bacteriophage DNA-dependent RNA polymerase to basically make genomic RNA mimics. And not having a cell culture system, the only way that would show infectivity was to actually inoculate this transcribed RNA into the liver of a chimpanzee, which did result in the initiation of replication. And at that time, we really thought that we were sort of on the home stretch because we basically had an unlimited quantity of infectious material that we could use to probe different cell culture systems to try and get this to replicate in cell culture. But that didn't turn out to be the case because transfecting these RNAs into various cell types did not lead to productive HCV replication or spread. So the next breakthrough came from Ralph Bartenschlager's lab, creating this guy called a subgenomic replicon. So this was a part of the HCV genome that had the 5' non-translated region and the 3' non-translated region, and then the structural protein coding region replaced with the gene for neomycin resistance, the NPT2 gene, an internal ribosome entry site from EMCV driving the believed replicase region of the viral polyprotein that was transfected into a human hepatoma cell line, these HU87 cells. And you could select with G418, and they showed that at very low frequency you could identify G418 resistant colonies that had persistently replicating HCV RNA in them. And so this was the first really robust cell-based system that we had to study hepatitis C replication. The problem was that this was a very inefficient process. So 1 in a million transfected cells could actually show initiation of replication. So it wasn't a robust genetic system for probing the function of the viral proteins and RNAs. And we actually identified the fact that these persistently replicating RNAs in these colonies actually harbored adaptive mutations. And when you could re-engineer those into sort of the original starting clone here and run through this transfection process, you could actually increase the efficiency of this by more than 50,000-fold. Single amino acid changes in the viral genome that could sort of yield this. So that was great. It really sort of provided a genetic system that we could begin to study these elements. And we could also produce cell lines that were very highly permissive for HCV. So sublines of these human hepatoma cells, the HU87 cells, HU87.5, so the HU87 lunate cells, when cured of replicons actually showed increased sort of ability to be transfected with these replicons and support HCV replication. So that was great, and it really seemed that the next step would be, okay, engineer these adaptive mutations into full-length genomes and find out if the complete life cycle of the virus was recapitulated. And when people did that, these RNAs replicated, but no virus was made. So again, there was another sort of block that sort of was not permitting us to sort of get to a complete replication system. And as I mentioned yesterday, the sort of breakthrough, again, 5 years later, and more than 15 years after the discovery of the virus, was this Japanese fulminant hepatitis isolate from Takagi Wakita. And this guy was able to replicate in these HU87 cells without adaptive mutation. So it was really the first example of this. And it produced virus particles that were infectious in vivo in the chimpanzee model and in liver humanized mice. So this really provided the first system to sort of study the complete life cycle of HCV. So the sort of mystery that loomed after this was, why don't most primary isolates replicate in cell culture? And why do we need these adaptive mutations? And so this was the sort of question that we sort of set out to ask. And, there are a couple of possibilities here. And one was, do these HU87 cells harbor some sort of inhibitory factor that or environment that blocks replication of these non-adapted isolates, or are they actually missing something? And I'd actually been trying somebody to tackle this project for a while. When Mohsen Saeed entered the lab as a postdoc, he actually came from Takashi Wakita's lab, so he was familiar with working with hepatitis C. And this is a picture of Mohsen in a happy moment, so he decided to sort of tackle this project. So like a lot of projects in the lab, we usually start with a bet. I was really betting that that these adaptive mutations were all over the place, and that since these adaptive mutations didn't map to sort of a single locus in the replicase proteins of HCV, that it must be that there are multiple ways to sort of change the structure of the replication complex to sort of overcome an inhibitory factor that might exist in these cells. And this just gives you an idea. This is from the early 2000s, just a map of the sort of non-structural polyprotein in the replicase that just shows you the spectrum of these adaptive mutations, some of which are more adaptive than others. So all kinds of different changes in this polyprotein can confer this sort of adaptive phenotype. So Mohsen disagreed with me, and he sort of took the alternative point that he thought there must be something missing. So I prevailed, and so he agreed to do some screens, but the first screen that he did, and since I'm trying to cover a fair bit of territory here, I'm not going to go through the gory details, was an shRNA screen. So we basically took these cells, transduced them with a lentivirus library that expressed a bunch of shRNAs, and then took that library and basically transfected that with a replicon, a selectable replicon, which didn't require one adaptive mutation. It required at least two adaptive mutations, so that the chances of getting two adaptive mutations in this initial transfection experiment were essentially zilch, and you shouldn't get anything unless you've really done something to the cells to promote replication. So Mohsen, even though he didn't believe in my hypothesis, he did this screen, and he was right. We got absolutely nothing. So as usual, I was wrong. So he got to do his screen, which was a gain-of-function screen. So he took a lentivirus cDNA library and used this to transduce HU87.5 cells. And we tried to do this at relatively low multiplicity so that there was just sort of a single cDNA expressed in each of these cells. This was a puro-selectable library, so we could select the cells with puromycin. And then the idea was that we would electroporate these cells with replicons that, again, required at least two adaptive mutations in order to replicate. And the hope was that we would get G418 resistant colonies popping up from the cDNA library, but not from the vector control. So he was right. He actually did this, and these are two replicons, a genotype 3A and 4A replicon of HCV. And you can see he did this sort of three biological replicates. You can see these beautiful G418 resistant colonies sort of growing up. And the sort of bottom blank lane is actually cells that have been transduced with a replicon that has a mutation that inactivates the RNA-dependent RNA polymerase of HCV. So this is a polymerase defective control. So what's in these cells? So he extracted RNA from 45 of these clones and sort of looked to see if the replicons in those clones actually harbored adaptive mutations that were wild type. And 76% of the replicon sequences in these clones were wild type. So they seemed to be replicating without adaptive mutations. There were some that were sort of a mixed population of wild type replicon sequences as well as mutations, and some that did have mutations, but those mutations didn't correspond to any of the previously identified adaptive mutations in HCV replicons. And we still don't know if they're adaptive or not, but obviously this looked pretty good in terms of establishing a system that would allow these guys to replicate without adaptive mutations. So, what are the sort of factors that gave rise to this? So when we amplified the cDNA ORFs, despite the fact that we tried to transduce these things with a low multiplicity, we got this awful looking mess of sort of multiple inserts in each of these clones. But there were three shown here that only had single bands. And Mohsen sequenced these, and they all corresponded to a cDNA encoding for a gene called Sec14L2. And well, what about the rest of these guys? It turns out that if you then make primers that are Sec14L2 specific and assay sort of all of these clones that were pulled out of the screen, basically the same gene is transduced in all of these permissive clones. So obviously that focused our attention on Sec14L2. So what is this guy? So it's been called C22OR6 or supernatant protein factor 1 or tocopherol-associated protein 1. That probably doesn't mean too much to most of you, but I'll come back to that later. It belongs to a family of lipid-binding proteins. It's ubiquitous but highly expressed in the liver, brain, and prostate. It has reduced expression in some kinds of cancer types. There are 3 splice isoforms, and it's mainly a non-nuclear protein. So is this really the smoking gun? I mean, we had to sort of prove it. So if you take, rather than these sort of library transduced clones, but you actually take a lentivirus that, excuse me, encodes Sec14L2 and transduce those, you can see that this allows, successful transduction of replicons for all of these different genotypes. So really, I think that proves that Sec14L2 transduction is responsible for this. It works in a number of different cell backgrounds, the sort of less permissive HLA-7 cells, as well as these Hep3B cells, which are permissive for HCV when you overexpress miR-122, what HCV requires to replicate. So what is it doing to enhance replication? Excuse me, sorry. So is it, these are some of the pathways that have been shown to be affected by Sec14L2. One is cholesterol biosynthesis, PI3 kinase inhibition. We checked those out. I'm not going to sort of show you all the gory details, but it doesn't appear that either of those pathways are sort of the responsible ones for enhancing HCV replication. And what about sort of vitamin E and other mechanisms? So what about vitamin E? So this has been called the tocopherol-associated protein. I didn't realize that tocopherol was actually vitamin E, but those are sort of one and the same. And there are some data that suggest that expression of Sec14L2 actually enhances the cellular uptake of vitamin E. And it turned out that when I was at a meeting while this work was in progress in Australia, I heard a talk by Stan Lemon, who talked about the work of Daisuke Yamane. And basically they had found that by supplementing vitamin E to cells, they could actually show enhanced HCV RNA replication. So this sort of got me thinking that these might be connected. And then this paper came out from the Lemon Lab, which basically sorry, shows that sort of if you look at the left panel here, and in red this is actually a genotype 1A replicon, which is stimulated by with a vitamin E addition in a dose-dependent manner. And interestingly enough, this JFH-derived replicon, the guy that can replicate without adaptive mutations, shown here in the white bars, is not stimulated at all by vitamin E addition. And you could actually mimic that using sphingosine kinase inhibitors as well. So both of these treatments, which were basically antioxidant treatments, seem to be able to enhance replication of the genotype 1A replicon, but not the JFH replicon. So we were curious as to whether or not overexpression of Sec14L2 might be phenocopying the effect of vitamin E. And it turns out that in many ways it does. So this shows you the enhancement of this replication of this genotype 1A replicon by Sec14L2 overexpression on the left, sort of about a threefold in this transient expression assay, which is very similar to what you see with vitamin E addition or with sphingosine kinase inhibitor. So again, sort of very similar in terms of the levels of enhancement. And so the question is, if you just add vitamin E to media, does that really sort of phenocopy the effect of Sec14L2? That's not the case, because as you can see here, this is what happens with these non-adapted replicons in Sec14L2 overexpressing cells. and vitamin E, although it may sort of enhance that slightly for this genotype 1b con 1 replicon, it really can't do that for these other guys. So there's something about stable expression of SEC14L2 that allows an environment where these wild-type replicons can persistently replicate. It does, however, actually, if you sort of combine the two, seem to make replication even better. So this just shows you an example of that with the same 3 replicons. So there really does seem to be some interaction between SEC14L2, and I didn't show you other stuff, but if you really look at sort of the additivity of this across different replicons, SEC14L2 tends to work better than vitamin E, and vitamin E will supplement sometimes replication of SEC14L2, but really we don't completely understand what's going on here yet. And I guess the sort of goal was, will the SEC14L2 expressing cells actually support replication of clinical isolates? And we're really just sort of getting into this, but these are 3 different sort of unselected patients here that we got from Andrea Branch at Mount Sinai. And you can see in the green bar that these guys can replicate in these cells. There's a reasonable signal-to-noise ratio, and that replication is blocked by the addition of an NS5A inhibitor to cladosphere. Sorry. So I've been talking too much today, I think. So at least for this part, we've taken this screen and identified this gene that can, when expressed, can allow replication of non-adapted HCV isolates. The replication efficiency still varies depending upon the HCV isolate and the cell background. So it doesn't really convert everything into sort of a miraculous high-level replicator like JFH1. It phenocopies vitamin E and the sphingosine kinase inhibitor in many respects. And it should really allow us to establish systems for these non-adapted sort of clinical isolates. So I think that's an example where we still sort of need to figure out the mechanism of this, but it's a significant advance for being able to propagate these wild isolates of HCV without adaptive mutations. So if I can continue to talk here without blowing your ears out from coughing. So I want to tell you a cautionary tale. So I think we're all sort of interested in the complexities of virus-host interactions, and there are sort of a number of ways that, and this I think applies to sort of any kind of functional interaction study. So we were interested in the NS5A protein of HCV, which is a component of the RNA replication machinery, as well as an important factor in assembly of infectious virus. And sort of one way to go about this is to basically try and look for NS5A interacting partners by IP mass spec. Sort of identify the potential interactors from those data sets. Do a functional screen to identify those that actually sort of show up as being important in your assay. Confirm the interaction. In the case of, I think, virus-host interactions, they're all interesting, but I guess if I have my choice, I like to study ones where, they're really sort of very close to a viral determinant. So a direct sort of viral protein, cellular protein interaction is a little bit easier to sort of dissect and understand. Sort of map the determinants, and then, sort of continue on and do mechanism of action studies. So, pretty standard stuff. You can sort of mix up the sort of way that you do these things. But so this was a project that a postdoc, Yo-Young Chung in the lab, was interested in. He did this, and he identified this protein, this NS5A interacting candidate protein MOBK1L1B. And this is a component of the hippotumor suppressor pathway. And without getting into the details of the pathway, we're sort of interested in this because some loss of function mutations in these MOB proteins in this pathway can actually give rise to certain kinds of cancers, including liver cancer. So it's kind of interesting in thinking about the possibility that an interaction or a sequestration of this protein by interaction with the virus might be a predisposing sort of pathway by which HCV was associated with liver cancer. So that was interesting. He basically sort of probed the potential importance of this with an siRNA knockdown experiment, which is shown here. So you can knock down this very effectively. You can get sort of significantly less virus infection and spread in this. So this really sort of seemed like we had a system with which to sort of study this interaction in a functional way in cell culture. So Joe Young is a fantastic biochemist, molecular biologist. He sort of went on to show using a yeast-2 hybrid screen that there was a direct interaction between NS5A and MOBK-L1B. And he sort of mapped that using sort of a series of deletion mutants in the yeast-2 hybrid system and also synthetic peptides to sort of map the NS5A region that was interacting with this. We determined the co-crystal structure of the complex to 2 angstroms and identified this tyrosine at position 317 and tryptophan at 312 that was important for this interaction. And then an interesting variant appeared. So this shows you in panel A the sequence of NS5A, and some of these sort of regions, and the sort of line in blue defines this minimal interacting peptide of NS5A with MOB KL1B. And so another lab, Heng Li Tang's lab, actually identified some binding sites for cyclophilin A. So cyclophilin A is an essential host factor for HCV replication. So HCV replication is inhibited by cyclosporin A. That was an early discovery that actually led to sort of a clinical development strategy, as you might imagine. And basically what Heng Li Tang's lab found was that there were a couple of changes in aspartic acid at 316 to glutamic acid, and this tyrosine at 317 to asparagine, which actually sort of released the requirement for cyclophilin A replication. So this was a cyclosporin A sort of resistant mutation. So this was interesting because it basically overlapped with the MOBK1 binding site that I just told you about on 5A. So we went ahead and explored this, and it turned out, as you can see here, and this is just one of many assays, that this resistance mutation actually abrogated at least this pull-down interaction between NS5A and MOB-KL1B. And on top of that, as you can see over here on the right, so this is wild-type HCV accumulating over time. And if wild-type HCV in a cell where you knock down cyclophilin A, replication is inhibited. In the case of this cyclophilin A resistant variant here shown in this bar, you can see that this guy replicates reasonably well, and it replicates just about the same in the knockdown experiment. But the surprising thing was that if you knock down MOBK-L1B with this guy, it actually knocks down the ability of this cyclophilin A resistant guy to replicate. So there was really kind of a disconnect there between the fact that it seemed like the interaction was abolished. Maybe there was somewhat lower level of replication with this cyclophilin A resistant variant, and yet the sort of knockdown experiment still demonstrated the phenotype of inhibiting this guy. So this got us a little bit suspicious, and actually you could explain this away by saying the pull-down assay really didn't reflect the actual ability of these guys to interact in a cell, and maybe it was a weaker interaction and the complex fell apart. And but anyway, the paper was actually written and accepted at this point, and there was a seminar at Sloan Kettering across the street from Eugen Buehler. And Eugen has actually run the sort of siRNA screening enterprise at Merck before they closed that down, and was now at NIH. And I didn't actually go to his seminar, but somebody from the lab came back and told me about this this C911 sort of control for siRNA screening. So I'm going to tell you a little bit about this. So, I mean, one of the problems with siRNA screening is that there are a lot of off-target effects. And so Eugen's sort of solution to this is schematized on this. So you've got your siRNA target, which also shares a seed sequence with a class of microRNAs. So in a in a perfect world, an off-target effect, this is going to hybridize to your target and lead to sort of degradation of your target RNA. But it's also going to sort of get incorporated into the RISC complex, and it's potentially going to interact with all of the microRNA target sites that sort of share that seed sequence. And so on the one hand, you know, these guys he came up with this C911 control where if you sort of flip these bases at positions 9 to 11, it creates a bulge. So you retain this potential seed sequence interaction. You have this bulge, and that's enough to sort of discourage the effect of these siRNAs from leading to target RNA degradation to having sort of a mild translational suppressive effect. And you know, the question is with that, I mean, you should get no silencing or diminished silencing of the target gene, but you will still maintain these interactions with sort of that you might be dysregulating with the microRNA population. So I thought, well, you know, we should go ahead and test this. And I have to give Yo-Young a lot of credit. He did this. And I'll just sort of show you, you know, some of the data from Eugen's paper. So on the left, this is a series of targets. This is actually a well-defined luciferase-based cell screening assay that he did. These are a number of guys where you can see the sort of extent of knockdown is really not influenced between the sort of original siRNA and the C911 control, whereas things that are really on-target effects, you can see that the C911 controls releases the suppressive effect of the siRNA and sort of gives you these varied levels of sort of restored expression. So we did that with MOBK1LB, and so this shows you an example of sort of the wild-type levels of this protein and the sort of replication of the virus with the sort of unmodified levels of MOBK1, and then siRNAs with different extents of depletion of MOBK1LB. And you can see that the one that has sort of better knockdown in terms of protein expression is sort of more you know, suppressive for HCV replication. So it looks like what everybody would like to see in one of these siRNA experiments. But when you do the C911 control, basically you can see that the C911s really show exactly the same phenotype on virus replication as the original siRNAs, despite the fact that they do not really sort of markedly knock down the protein, shown here with the C911 controls. So, you know, I just throw this out there as a sort of cautionary note. Probably more than 80% of the hits that you get on siRNA screens are off-target effects. And it's one of the reasons why when people did screens for HIV host factors, and 2 or 3 labs did these, there was essentially no overlap in the hits. If you sort of check out some of Eugene's publications on this, he's actually taken different siRNA labels, targeting overlapping sets of target genes, screened them against the same assay, and actually found absolutely no convergence of the hits between those libraries. So the lesson learned is to beware of siRNA results. Just be, you know, really skeptical and set a high bar for functional validation. I think, you know, we did do a lot of the controls, but, you know, sometimes when the controls didn't work as the way we, you know, sort of expected them to work, we could wave our hands and, you know, sort of explain them away. And, you know, really in terms of science, be your own worst skeptic. I mean, you've got to think, you know, why this result might not be what you think it is. It's really, you know, very important. And why might your, you know, favorite interpretation be wrong? So anyway, that's my sort of quick sort of cautionary tale. So how does interferon actually work? So as I mentioned before, we got interested in this really because of our work on HCV and trying to understand, what it might take in terms of antiviral development to actually get rid of interferon. So I think many of you are familiar with all these innate immune response pathways. So we've got, various PAMPs that are sensed by either toll-like receptors or some of these intracellular pattern recognition receptors like MDA5 and RIG-I. Those sort of signal for the assembly of transcription factors like IRF3 and NF-kappa B that head into the nucleus and turn on sort of various genes, either directly regulated antiviral genes or interferons, which could then secreted and bind back to sort of receptors. They can be type 1 or type 3 interferons. This is a somewhat old type 3 interferon scheme. And then that basically turns on this remarkable JAK-STAT pathway that turns on a spectrum of interferon-stimulated genes and that have, various antiviral effects. And we know how some of these guys actually function. So John Scoggins, when he showed up in the lab, I'd actually been trying to get somebody to do this project for about 10 years. And, I would take people to the faculty club at Rockefeller and start pouring lots of beer and wine down them. And then I'd sort of ask them, there must be a lot of interesting sort of interferon-stimulated genes whose functions we don't understand. Would you be interested in tackling this project? And I had failed miserably every time. And so I tried this on John Scoggins, who was a graduate student at Cornell, and sort of went down to the faculty club, and I found out that he didn't drink. So this was kind of a setback. But when I suggested that this might be an interesting project, he said, "Oh yeah, let's go for it." So there are hundreds of IS genes that are induced by interferon, sort of different spectrum of genes and different cell types under different pathogen infection conditions. And so we really sort of became interested in trying to understand more about how these interferon-induced effectors might work against a spectrum of different viruses. Would there be, common themes, so that if you found a gene that inhibited flavivirus replication, would that work against all flaviviruses, or were these species-specific, genus-specific, family-specific, and things like that? So this was a bad idea for sort of many reasons. I'll just sort of go through those now. First of all, it's probably bad for us as molecular virologists to think that we could, really begin to study interferon. This is such a complicated system. It's really a remarkably effective system, and so it's likely redundant. So it may be very difficult to sort of pull out things that have really marked viral specificity. And of course, that could be one of interferon's secrets of success. It's basically nature's sort of broad spectrum combination antiviral therapy. We sort of decided, because of the redundant argument, that, well, maybe we should take an ectopic sort of overexpression strategy. But of course, this is going to give rise to all kinds of false negatives, because some of these interferon-induced genes really are probably going to need a constellation of other factors that are induced by interferon in order to function. We're going to get false positive by overexpressing these proteins out of context. The other sort of argument for not doing it is that, again, really sort of getting the right cellular context, the right virus, sort of mesh. If you're working with the human ISG library, I mean, what forces in evolution have shaped this constellation of interferon-stimulated genes? We probably don't even know what most of these pathogens are today. And then, of course, the other thing is that this kind of a fishing expedition is unfundable. So with these sort of thoughts in mind, we decided to do it anyway. And so John spent a lot of time optimizing lentivirus vector for expressing these ISGs. I won't go through the details. Actually, to get sort of balanced expression of this bicistronic vector is not trivial. It's marked by expression of a red fluorescent protein. And basically, the screen was really pretty simple. You take a lentivirus that expresses a control, like firefly luciferase, for example, or an ISG. make a transduced cell population. So the productively transduced cells are going to light up and be red. And then we sort of biased and made our lives easy by using viruses that express GFP. So we could look for the frequency of double positive cells, basically. And if it inhibited the ability of this virus to replicate, we would sort of see a lower frequency. So this would be what we would see with the control, and a lower double positive population, or a lower level of GFP expression if replication expression was impaired. And this just shows you what this looks like for yellow fever. Looking at a firefly luciferase control here, and also IFI6, which is actually an interferon-stimulated gene that inhibits flaviviruses, so you can see a clearly diminished signal. So John went through sort of all the databases on interferon-induced genes and came up with a list of about 450 of these. And so we sort of narrowed that down to a smaller set of 387 of these guys, and built sequence-verified bicistronic vectors for all these guys. Use these to rescue lentivirus stocks, as sort of shown here. And one thing that you'll notice in this sort of in-your-face RFP multi-panel figure is that some of these actually produce really nice sort of RFP lentivirus titers, and others are just kind of black holes. And I won't have time to go into this today, but John was initially really upset about this, because I think about 40 or 50 of the genes we didn't get very good lentivirus titers to actually be able to do the screen. But when we looked at these guys sort of down here with these sort of very low lentivirus transduction efficiencies, we found in there that there were things like ApoBec3G, tetherin, and other things that had been known to basically inhibit HIV. This actually, in collaboration with Paul Binoch's lab at the Aaron Diamond, provided quite a sort of rich source of new ISGs that inhibit HIV replication. So the sort of initial distress associated with not being able to study these things by lentivirus transduction actually turned out to be a bit of a gold mine in the end. So if you do this screen against a variety of viruses, we did this in our favorite hepatoma cells, H287 cells for HCV, because obviously you're limited to some extent by the cell type that your virus will infect. We used MT4 cells for HIV, and then a couple of flaviviruses, yellow fever, West Nile, and a couple of alpha viruses, Venezuelan equine and chikungunya. We actually did those screens in STAT1 fibroblasts, because obviously one of the things that we didn't necessarily want to sort of pick up here were, say, transcription factors or other things that just induced interferon production, fed back into the cells, and then turned on all the ISGs. So this just sort of gives you a plot of these 300-odd genes, and there are some highlights here. So overexpression of the transcription factor, interferon regulatory factor 1. This actually, in just about every viral context that we've checked, this actually exerts a subviral, or actually a sub-interferon program of interferon-stimulated genes that inhibit just about every virus that we've looked at so far, RNA virus and DNA virus. And then one of the other guys was this protein that very little is known about, or was known about, C6ORF150. And we found not only genes that had very marked sort of antiviral properties, but interestingly, some of these genes, when expressed alone, actually showed proviral effects, like ADAR, double-stranded RNA-activated adenosine deaminase. So if you look at a word cloud of these initial results, sort of based on the sort of level of ambition, the sort of big winner, IRF1, C6ORF150, some of the pattern recognition receptors, RIG-I and MDA5. And I guess with some of the mechanistic studies that John did showed that actually early steps in virus infection tended to be targeted by a lot of these ISGs, translation. So translation is worth studying. As I said, IRF1 establishes an antiviral program interferon of interferon signaling, and this new molecule, C6ORF150, that had broad antiviral activity against these positive strand RNA viruses. So we wanted to look at this a little bit more broadly. So we expanded the screen to include some additional viruses shown here. Vaccinia, Coxsackie, poliovirus, other alpha viruses shown here, some of the negative strand viruses, paraflu, NDV, RSV, measles virus, and Abunia virus. And again, we could find genes that had sort of very strong antiviral activity. And interestingly enough, if you just sort of cluster these antiviral ISGs in a sort of very simple clustering analysis as to sort of which ones are effective against which viruses, they actually do segregate into sort of positive strand RNA viruses and negative strand RNA viruses. In general, the negative strand RNA viruses seem to be much more resistant to sort of the expression of these ISGs. So it may be that in order to get more effective sort of negative strand virus inhibition, you need, sort of more than one of these guys expressed at the same time. We've done some of those combinatorial experiments, but I won't have time to go into them today. So again, in this more broad screen, this guy, C6orf150, showed up again. And so we became kind of interested in this guy, and I just want to sort of take you through sort of what's happened there. So C6orf150 has this MAB21 domain, which is a C. elegans protein involved in cell fate determination. It is predicted using bioinformatics to be a member of the nucleotide transferase superfamily, and that includes enzymes like 2,5-OAS, another sort of interferon-stimulated gene, RNA capping, poly-A polymerase, and so on and so forth. So we became interested in this, and John, sort of began to, beat the protein up. We didn't have a structure of this protein. There's a predicted disorder region at the N-terminus, and then various sort of structured either beta sheet or alpha helical regions, and then some predicted catalytic residues in this nucleotidal transferase activity that are shown here with asterisks. And so John made a series of deletion analyses, and the bottom line is that really the only thing that we could sort of delete and still retain functional activity was this N-terminal sort of disordered region shown here. All the rest of these deletions were defective. And similarly, if you mutate these residues that are predicted to be associated with nucleotidal transferase activity, these guys are also sort of loss of function in terms of its antiviral activity. So, so we were working on this when sort of two papers appeared in Science back to back from Zijin James Chen's lab at UT Southwestern. Now this was a place where actually John, by that time, had moved to be a faculty member, and C6orf150 was going to be his, sort of fish to fry as a starting faculty member. And these papers appeared about 6 months after he arrived there, and they're really, if you want to read a couple of amazing sort of biochemistry papers, these would sort of fall in that category. And so James was sort of interested in trying to understand more about how DNA was sensed and turned on some of these innate immune responses. And basically he had discovered that there was an enzyme, this cyclic GMP-AMP synthase, which basically made a cyclic dinucleotide that was able to sort of turn on interferon in a sting-dependent way. So basically, cGAS, this cyclic GMP-AMP synthase, binds DNA. It sort of makes this cyclic dinucleotide, which then binds to sting and then activates IRF3 in ways that we don't understand yet. And then IRF3 can either directly turn on sort of antiviral genes or interferon. So this really a couple of fantastic papers. This was just sort of really brute force biochemistry in terms of setting up the assays to do that, purifying this small molecule, which would sort of elicit this reaction, and then figuring out the enzyme that was responsible for making it. So it turned out that if you sort of looked deep into the Chen Lab papers, that yes, indeed, cGAS was actually C6orf150. So the sort of probing from our standpoint, and some of the data that we had at that time was that if you just overexpress this in cells, C6orf150 was able to basically activate a subfraction of interferon-stimulated genes sort of directly in the STAT1 defective fibroblasts. And it elicits this IRF3-driven antiviral program, again, sort of independent of interferon signaling. This is a STING-dependent process. So it turns out that STAT1 defective fibroblasts have STING, and you can see this antiviral effect of overexpressing cGAS for Venezuelan equine encephalitis virus. But for HU87 cells, interestingly enough, for reasons that we don't understand, they don't have STING, so you don't see this effect when you overexpress cGAS. So it is a STING-dependent effect. This is just another experiment where we've used siRNA to sort of knock down STING. And you can see here that, again, in the STAT1 fibroblasts where you overexpress cGAS, you get activation of IRF3, and presumably this is the pathway that leads to the stimulation of these interferon-stimulated genes. So again, in our hands, STING is required for this, this antiviral program. So the question is, why is a DNA sensor, sort of, why did we pull this out, in an RNA virus screen? And I guess the thing that we had discovered, and that again sort of the Chen lab published sort of before we did, was that actually HIV and this lentivirus delivery system that we were using to express cGAS in these cells was actually the trigger that was stimulating cGAS to make cGMP and stimulate string and turn on this antiviral program. So really sort of quite an interesting and I think unexpected result. So in the meantime, we were making knockout mice, and we sort of decided many years ago, before we even knew what this was doing, to make these animals in collaboration with Skip Virgin. And this just shows you, I won't go through it, the sort of characterization of some of these mice. And interestingly enough, as you might expect, these show sort of a remarkable sensitivity to DNA viruses like vaccinia virus. And you can see with MHV, a herpes virus infection of macrophages, this dramatic stimulation of interferon beta messenger RNA in the wild-type mice, but not in the STING knockouts. So again, that sort of fits with the model. What is still, I think, quite surprising, and what we found was that these mice still exert a sort of a hypersusceptibility to infection with an RNA virus like West Nile virus. And we still, to this day, really don't understand what's going on here. So, if it's a DNA sensor, how does it play a role in RNA virus infection in vivo? And is this due to some sort of heightened innate immune activation that sort of occurs if you have cGAS present that confers some sort of innate level of protection? Or is it that this, cytopathic RNA virus is actually, liberating host DNA that can then serve as a PAMP to activate cGAS and STING and IRF3? So that's something that is still to be determined. So this really is a field that moves pretty quickly. So just to give you a timeline of that is not complete. There are probably another 20 or 30 publications since this slide was made. But we pulled this thing out in 2009, and it was pretty clear at the outset that this was sort of related to 2,5-OAS, and we sort of connected with Skip Virgin in 2010 and started making these mice and sort of published this antiviral activity that it had in 2011. And then the sort of Chen papers came out at the end of 2012. Then the field really exploded with a lot of structural biology. We now have structures for cGAS in complex with DNA, with STING in complex with cGAMP, the cyclic dinucleotide. Lots of labs. This all unfolded really within a 4-month period from about 3 or 4 different labs. The fact that it's a sensor not only for the DNA viruses, but also for HIV and the knockout mice. So it's really a story which I think is, again, a reflection of sort of how much we don't know about these innate immune responses, because, it turns out that, this is really an important pathway that influences, the whole sort of field of DNA vaccination, for example. James was at Rockefeller a couple weeks ago giving a talk. And if you look at sort of the vaccine efficacy in in the C-gas defective mice with the DNA vaccination protocol, absolutely nothing. So the sort of adjuvant effect of DNA is very important for getting a sort of a DNA vaccine-driven immune response. So anyway, that's sort of a fishing expedition that, at least for us, I think, ended up catching some interesting fish. It's the sort of thing that NIH will not fund. These are in order to sort of start these projects. You need to have some discretionary money, or go rob a bank, or convince your department chair to fund it or something, which is obviously getting harder to do. I mean, I think if we couldn't really do this today, although the situation was a little bit different 6 or 7 years ago when we started this. So I think there's still a lot to do, and to me, interferon is still this amazing sort of biological control system. And I think some of the things that we're trying to do in a relatively comprehensive way is to look at all of these different aspects of the interferon response using all of these different technologies that we have. And it's really going to be a challenge to sort of figure out, I think, how nature has evolved this system to be able to deal with this amazing spectrum of pathogens, and in a way that, when a cell is treated with interferon, it induces all these proteins, this amazingly sort of broad spectrum antiviral state. And if that cell, is not pushed over the edge by a viral infection, the whole thing sort of shuts down, auto-regulates, and things return, I don't want to say back to normal, but the cell doesn't die, and yeah, things pretty much return back to normal. So what is the cell biology that is, sort of associated with that amazing reorganization that poises these cells to sort of resist a viral invader? And how does this actually unfold in 2 and 3 dimensions in vivo? Because obviously these things are cytokines that are going to have sort of local and diffusion limited effects. So those are the 3 stories. I'd like to point out John Scoggins. He's down here, who started this work in our group. Really an amazing scientist. I mean, you can imagine just sort of keeping track of all this on this scale is no simple feat. And we really had, everybody, a lot of people in the virology field either contributed viruses or helped us develop assays to do screens with their viruses. So really quite a group effort. We had some initial funding from one of these regional centers of excellence, the sort of Northeast Biodefense Center. Some of this money that was poured into questionable research after 9/11, but some of it got recycled, hopefully for a good purpose. And the other stories that I told you about, Mohsen Saeed, sort of on the SEC14L2 study, he's also a really fantastic virologist, and Yo-Young Chung, who obviously, his project didn't turn out the way that he wanted it to, but I really admire this guy for, sort of pushing through and, basically, proving that the whole thing that he'd been working on for 2 to 3 years was an artifact. It takes a lot of guts to do that. We sort of withdrew the paper from the sort of higher profile journal that it was in, and did publish a JVI paper sort of telling this lesson learned story. But, it just shows you that, you've got to be sort of very careful. And again, he did an amazing job on this, and has got some other good projects cooking. But so that's it. And if anybody has any questions about these short stories, I'd be happy to try and answer them. Thanks. No? Yes.
yesterday's lecture and today's both were really, really nice. Just a couple questions on the C-gas. I was just wondering, and these are sort of obvious sort of questions. Has C-gas turned up on any GWAS studies or other sort of?
I think there are some sort of polymorphisms in C-gas. I'm not sure they've really sort of been explored yet in terms of disease association, but yeah.
Another question on that. Has anybody looked on the effect of C-gas with some intracellular bacteria, cell or something like that?
No, not to my knowledge. I mean, that would be something that would be, very, very interesting to look at and see if that's sort of in the pathway. I mean, I think that there is some work, not by us, but again by James's lab, sort of looking at MTB. And again, it seems to be very important in the sort of host response to that.
One other question.
Yeah.
Too. I may have missed some of the subtlety, but on the siRNA screen that you did, even though the initially identified target wasn't correct, there's probably still something in there.
Oh yeah, no, absolutely. Yeah, we really haven't sort of made an attempt to sort of figure out what the off-target target was. But I mean, that is, at that point, I think we were just kind of feeling a little downtrodden and beaten. But that can be done to some extent informatically if you have sort of broad enough siRNA screening data, because you can begin to sort of look at the phenotype that you see and sort of match that with different seed sequences and so on to try and identify potential targets. But you're absolutely right, and actually Eugen has sort of created some software to do that kind of analysis. We just didn't have a sort of a deep enough siRNA screening database to look at that. The audience is stunned. Okay, all right, thank you.